Hey, sweetest potatoes. So, in the previous online class, we talked about phototropism, which is a physiological phenomenon in plants where the plant bends or grows directionally in response to light. And we also talked about the experiments which were carried out, which led to the discovery of phototropism. The experiments which were carried out by Charles Darwin in the year 1880s, where he told a signal was involved, and later in the year 1930s, Peter Boyson and Jensen called this chemical as auxin. We have come a long way since the initiation of Darwin's substance, auxin, as a major regulator of phototropic responsiveness, and despite the seemingly physiological simplicity of phototropism, it continues to amaze us with its complexity. And moving forward, for now, we know that phototropins and auxins are the main players which are involved in phototropism. And in those phototropins, there are mainly two phototropins which regulate or which are involved in phototropism. That is, you have phot1 and phot2. So when there is a low light condition, so when there is a low light condition, the phot1 gets activated and it triggers the phot2, which redundantly stimulate the chloroplast positioning along the cellular edges which are perpendicular to the incident light. For example, if you have the incident light coming in this way, all the chloroplast is positioned perpendicular to the incident light along the edges. But if there is a high intensity of light, when there is a high intensity of light, then the phototropin phot2 gets triggered or phot2 gets activated, which makes the chloroplast move away from the edges of the cell to avoid photo damage. Like they say, too much of anything is bad. So when there is too much of light, it's like a uh, protective measurement that this plant takes with the help of this phototropin phot2. Coming to the processing of light cues. So though we have a very seemingly simple physiological phenomenon called as phototropism, the process or it is very complex, and we have been just figuring out puzzles of this whole phenomenon. So coming to this processing of light cues, there are many players which are involved in this process. So there are about 6 adaptive photoreceptors which are involved, which includes phot1, phot2, phytochrome A, phytochrome B, cryptochrome 1, and cryptochrome 2. There are also many primary directional BL receptors and signaling complex, like you have that there are multi protein families which include phot1, phot2, NPH3, RTPT1, SCF complex, PKS, and ABCB19. And there's more. There are about 9 auxin transporters. To name a few, you have pin 1, pin 2, pin 4, pin 7, and ABCB19. There are 7 auxin receptors, and there are few extracellular receptors which includes ABP1 and intracellular receptors like TR1, AFP1, 5. And there are also many gene expression pathways which involves transcriptional regulators. Like there are a lot of them. To name a few, you have NPH4, ARF1, ARF19, MSG2, IAA19, IAA29, and so on and so forth. And there are also many target genes which are involved in this process of phototropism. To name a few, you have EXP1, EXP8, and so on. And there is also integration of at least 4 phytochrome signaling pathways, and also many hormones which are involved like auxin, ethylene, BR, and gibberellins. So although we just know the list of these associated complexes continue to grow, so does our understanding about the functions of these components and their interactions. Fortunately, the phototropism path before from the time like before the Darwin to date has been continuous, and it has been populated with creative individuals, and it is just a matter of time before the phototropic response puzzle is revealed, and we put all the pieces together to get a complete picture. So I hope you understood this concept of phototropism, what we know, and there is so much more to this, and all the references used for this video has been linked in the description. And if you like this video, consider subscribing and hit the like button, which will inspire us to make more. And thank you so much.